Hey there, biologists! In this set of notes, we are going to explore gene mutations and the things that cause them, which are called mutagens. So let's start with the types of mutations that exist and how they impact protein function. So, classification of mutations by their effects on DNA. Gene mutation is defined as one individual gene is affected by a mutation. There are three types. One is substitution, where one or more bases are replaced by another. If you take a look at this original DNA sequence, we have 3 CG base pairs followed by 3 TA base pairs. And in a substitution mutation, we have at least one or more of those that are substituted for another type. So in this case, we have an AT where there should be a CG. A deletion mutation is when one or more bases are deleted from the sequence. In this image, you can see that one base pair was removed. An insertion is when one or more bases are inserted or added. Pretty simple and straightforward. There are other types of mutations that exist, and we will eventually talk about them. These are chromosomal mutations, and what this means is that we're talking about the impact of a gene mutation on dozens to hundreds of possible genes. So this is where you might have an entire section of a chromosome that contains dozens or hundreds of genes that is completely deleted, or maybe that section of the chromosome is accidentally added on to an entirely different chromosome. Or maybe an entire section of a chromosome is doubled and duplicated, or maybe it's flipped and inverted. We will talk about these types of mutations next unit. So hang tight on this idea. We will talk about what happens when many genes are impacted at one time. But for today, we're going to be focusing on simple gene mutations, how one specific protein sequence is affected, whereas this focus is on how our entire sections or many proteins affected by a mutation. When do gene mutations occur in the life cycle of a cell? Well, they have to occur before or during the process of DNA replication. So remember that a cell is born and enters its life in gap 1, where it grows and does its celly jobs. And then if that cell is prompted and needed for cell division, that's when the DNA will be replicated. So perhaps during gap 1, maybe the cell was exposed to too much sun UV radiation, and maybe the mutation happened during this cycle. Or maybe when the DNA was being copied, maybe a DNA base pair was accidentally put in the wrong place or deleted. So this process can happen both before and during the process of DNA replication during the S phase of the cell cycle. And what that means is that if this cell doesn't correct that mistake and DNA successfully replicates and the cell successfully divides, that means that every cell that comes after with that mutation will be impacted. So this cell will divide to form 2 and then 4 and then 8 cells and so on and so forth, and all of the cells thereafter will inherit that mutation. Now I want to think big here. Each of us started out as one single fertilized cell, an egg cell that had been fertilized by a sperm. And so we started out as one cell, and we are now the product of trillions of cell divisions. So if you think about it, if this process happened when you were just one cell or a handful of cells, then that means every cell in the body inherits that mutation. Whereas if this mutation happens later in an organism's lifespan, the impact would be lessened. So think about it in the big scope too. So consider the following scenarios and state whether the mutation is likely to be passed on to the organism's offspring and be able to explain why. So let's read through each one. I'll give you some thought time and then we'll talk about the answers. So A, a single bacterial cell contains a mutation in its DNA. B, a skin cell on a cat contains a mutation. Or C, a sperm cell in a whale contains a mutation. So think big about bacteria, skin cells on cats, and sperm cells in whales. And think about it. Let's check our understanding. So if a bacterial cell contains a mutation, well, remember that bacterial cells replicate by simply copying themselves. So yes, every cell that comes after that original mutation will inherit that mutation. Do cats reproduce using their skin cells? No. So if a cat has a skin cell mutation, that does not necessarily mean its offspring will inherit that mutation too. What about sperm cells in a whale that contain a mutation? Well, if that whale does successfully reproduce and that sperm cell is the one that's used to fertilize an egg, then yes, that could potentially be passed on to its offspring. Outcomes of gene mutations. Let's talk about some basic vocab. So we are going to be using the terminology called reading frame. And a reading frame refers to the codons on the mRNA that are read in groups of 3 letters at a time. So this reading frame has 1, 2, 3, 4, 5 codons, and each codon has 3 mRNA bases. So depending on the type of mutation, it may or may not impact the reading frame or the codons. If the reading frame does not change because of that mutation, then the impact is lessened. If the reading frame is affected by a mutation, then the impact is greater, and we'll talk about why coming up. So this triplet nature of the genetic code is really essential to maintain, and that's because remember that 3 letters at a time are read by ribosomes, and each one codes for amino acids. So if we alter or change these amino acids in any way, it may have a great impact on the amino acids that it codes for. So let's talk about how this works. So I want you to think about it first. Which types of gene mutation do you think will change the reading, or will not change the reading frame below? So would a substitution, deletion, or insertion mutation not change this? And which types of mutations will change this? Think about it before we check our answers. All right, let's see how you did. Well, if you think about it, a substitution mutation, well, if we change one letter, it doesn't change the rest of these. We still have 3 letters for each of these. Whereas if we delete a letter or insert a letter, that means that we're either shifting forward all of the bases to reform those groups of 3, or we're pushing letters backwards to maintain those groupings of 3. Let's see what that looks like. So here's our original mRNA sequence. A substitution, we've changed one nucleotide, but all of these others remain the same. So we have not shifted the reading frame. So this is less of an impact in comparison to the others. In this sequence, we deleted a C. We deleted this 5th we deleted the 5th base there. And so what that means is that all of the bases after are going to be read by the ribosome 1 earlier, again, to maintain those groupings of 3. In this case, this 3rd codon happened to stay the same, but all of the other codons afterwards were impacted. An insertion mutation, here we added an extra C. And again, our 3rd codon happens to still remain AAA, but all of the other codons after have now also changed. So in deletion and in insertion mutations, we are shifting our reading frame. So therefore, when we think about how we classify these, if the reading frame is unchanged, then it is one of 3 types of substitution mutations, which we are going to talk about next. So again, a substitution mutation, we change 1 or more bases, but the reading frame is intact. Whereas if the reading frame is changed, this is called a frame shift mutation, and this is due to deletion or insertion of 1 or more nucleotides. Let's talk a little bit more about these different types of substitution mutations. So again, substitution mutations exchange 1 or more nucleotides for another. A substitution mutation can also be classified as a missense substitution mutation if the amino acid that it's supposed to code for has changed to another amino acid. So in this example, we see all of these bases are cat, and then the RNA would be read. So this is a little bit misinterpreted. Remember that RNA is what would code for these, so this has been simplified. So all of these amino acids are histidine, or his. And in this case, we see that CAT has been mutated to be CCT, which means that this now codes for an entirely different amino acid. So what this means is that this is a missense. 1 amino acid has changed. And because most of this protein sequence is intact, the protein may still function relatively normally. It may not function at all. It may take on a new, entirely new function, and we'll talk about some of those examples coming up. But this impact is significantly less in comparison to the others we will talk about. Oh, well, except for one other. All right, so a nonsense mutation. This one's easy to remember because think about no as in stop. So this is when an amino acid has been changed to a stop codon. So here we're supposed to have 1, 2, 3, 4, 5, 6, 7. We have at least 7 amino acids in the sequence, and you can see that it's supposed to keep continuing. But this substitution mutation has artificially inserted an early stop codon. So that means that our protein is significantly shorter than it's supposed to be. Now this could perhaps happen at the second to last amino acid, so maybe it's not that significant of an impact. Or maybe it happens towards the very beginning of that protein sequence. But either way, this early stop codon may or may not have a really severe impact on whether or not that protein will function at all. Obviously, if that change happens later in the sequence, the protein may still function relatively okay. It just depends on the case. And last, this one's a lot really easy too. Similar to no as in stop, silent means that a substitution mutation occurred, but the amino acid that it codes for is still the same as the original sequence. So in this example, we see we're supposed to have methionine, lysine, phenylalanine, and glycine, met, lys, phi, gly. And here we can see that we have AUG, AAG, that's the same, UUU, oh, here's our mutation, GGC has been changed to GGU. Now, remember your codon chart. Remember that every amino acid has multiple codons that may code for it. So sometimes a substitution mutation will still produce the same amino acid. So this is called silent because even though that mutation is there, it's silent or has no effect on the protein because it's the same amino acid that it codes for. So that's, if you're going to have a mutation occur, that's not a bad one to have because you still have the same protein. So again, think silent as in you don't notice it's there because it produces the same sequence. No as in stop. Early stop codon in missense kind of partially doesn't make sense. Okay. Oh, and here's that codon chart. So again, remember that, if you had a CCC mutated to CCU, that still codes for proline, so that would be a silent mutation, same amino acid. Just a review of the frame shift mutations. This is another way to visualize the deletion and insertion. So deletion, when we delete even just a single nucleotide, it potentially changes every sequence that comes after. So significant impact. And then same thing with insertion, where you have every amino acid after that dramatically changed. So this is kind of just a review recap. Let's see, mutations outside the gene coding sequence can also impact gene expression. Okay, so we've been talking about DNA sequences and how we've learned that there are parts of DNA that code for a specific protein. And outside of those genes are other parts of DNA beyond, before, and after a specific gene for a protein. And we had to cut some of this content this year because of, we're just reducing content, but there are parts of the DNA outside of a specific protein coding gene sequence that may or may not have impacts on protein function if it's mutated. So some sections of DNA are not impacted at all if there's a mutation. So we actually have tons of mutations in our genome that have no impact on our cell function and protein function. Whereas if a mutation happens in a region of DNA called a promoter or terminator, which are important regions for turning on this process of protein synthesis, and some of these other things that we didn't have a chance to talk about. So sometimes even though a mutation happens outside of a gene, it can still impact gene expression and how protein synthesis works. This is a little bit beyond than what we got to this year, unfortunately. Mutations can also be classified by how they impact protein function. So some gene mutations are characterized in, when we talk about genetic diseases eventually, some are called loss of function mutations. And this means that a mutation has resulted in a complete loss of function where the protein doesn't work at all, or a reduction of function where the protein doesn't work as well or less of the protein is made. We're going to talk about a couple of examples coming up in a minute. And what's really interesting about mutations is that some mutations can result in a gain of function, where maybe something works better than the original gene sequence did, or maybe it has an entirely different function altogether. So sometimes that's good and sometimes that's bad. Let's talk about some cool examples. This is a really important reminder. So starting now especially, and then moving forward in the next couple of units, we're going to be talking about many case studies of human diseases and disorders. And when we do this exploration, we're going to be looking at descriptions and images that show things that we may be unfamiliar with and may sometimes be graphic in nature because they display medical information. So when we are talking about genetic diseases and disorders, it is absolutely critical that you approach this with a mature and respectful mindset. So we need to keep in mind that some of our classmates, friends, and family members may in fact have some of the diseases we will be learning and talking about. So it's okay to ask questions, it's okay to be curious. I really invite that and encourage that, but it is not okay to laugh or criticize or make jokes about something that we're learning about and seeing. So laughter is not appropriate, okay? So here's one example of a gene mutation that causes a disease called neurofibromatosis. And this is caused when neurofibromin, which is a protein that regulates another protein, which regulates cell division. So basically think of it like a domino. You have one domino that turns on another domino that's supposed to control how much cells divide. Well, the gene that controls this toggle or this switch is very long. There's over 8,000 A's, T's, C's, and G's in that DNA gene. And so therefore it's really likely that if it's a big gene or a long gene with many bases, it's more likely that a mutation will happen at some point in a family's history. And if that gene gets mutated, then we're losing a switch. And therefore it means that the process of cell division is rapidly sped up, in people who have neurofibromatosis. So this is one example of a loss of function. The protein has lost its function in controlling cell division. Another example that we learned about in our explo, our gene mutation explo, is cystic fibrosis. So cystic fibrosis is a disease that's characterized by the dysfunction or loss of function of a transport protein. There's a transport protein that's supposed to help move salts in and out of cells. And in people with CFTR, they might have a completely non-functional protein or it's completely missing. And in some cases, the protein is there, but maybe it's just not working as fast. So, and again, this is another example of a really long gene. So CFTR cystic fibrosis is a relatively common disease in comparison to others. And the current average lifespan for cystic fibrosis is around 44 to 45 years of age. But it varies depending, again, on how severe someone's mutation has been. If it's really severe, maybe that protein doesn't work at all. Unfortunately, significantly reduced lifespan in comparison to if the mutation still produces a relatively functional protein, but maybe just doesn't work as well. Lactose intolerance, this is a cool one. This is an example of a loss of function mutation. So did you know that all other mammals cannot easily consume milk after infancy? And in fact, the majority of the human population, when you think about the 9 billion people on this planet, the majority of those people cannot easily digest milk products as an adult. And this is because there's a gene that's supposed to turn off after infancy. And so people without these changes have the reduced ability to digest lactose as they get older. We'll talk a lot about lactose intolerance coming up. Here's an example of a gain of function in a negative way. So this genetic disease causes hereditary pancreatitis, which is caused by a mutation that causes a digestive enzyme to become more active. So it's a gain of function because the enzyme is more active, but it causes a bad disease. So just because something means gain of function does not mean it's always going to be a good thing. Excuse me. If someone inherits one normal copy of a gene for sickle cell anemia and one mutated copy, they are resistant to malaria. So this is an example of a gain of function in both a good way and a bad way, because if you inherit 2 mutated copies, then you have sickle cell anemia and you are likely to get blood clots and die. But if you have 1 mutated copy and 1 healthy copy, you are immune to malaria, which is very common in tropical areas in the world. We will talk a lot about this coming up this year. Pretty interesting stuff. So in a nutshell, it's really important to recognize that mutations are not always bad. Mutations, for example, may occur in a region of DNA that does not code for a gene or does not provide any kind of function. It's just what scientists have kind of called junk DNA, which is kind of a term that's not widely used anymore. But mutations may also result in a trait that has a neutral effect. All of our eye colors are the result of gene mutations. So that doesn't affect our survival, it just happens to introduce new traits. Pretty cool. And mutations may introduce new advantageous traits. For example, sickle cell anemia provides malaria resistance. There are tons of other types we are going to talk about coming up. There was another assignment that we did last year that we had to skip this year, where you would have learned, for example, that there is a group of people who live in the Himalayan mountains who have a gene mutation that increases their ability to essentially carry oxygen through their blood in an environment where there's reduced oxygen concentrations. So pretty cool. That is a gain of function. Awesome. All right, we'll talk a lot more about those coming up. So we've learned that gene mutations are caused before or during DNA replication, and this is because of a couple things. It could be bad luck. Sometimes a spontaneous mutation just happens. But otherwise, those mutations may be caused by things that we're exposed to in our environment. I mentioned, like, for example, if you soak up too much UV radiation in your skin cells, you are more likely to mutate your DNA in your skin cells. Therefore, that's a mutagen. In comparison to, if you were to eat healthy foods, that reduces your chances of having a mutagen because you're getting all the essential amino acids and vitamins that you need. So mutagens can be environmental, and sometimes mutations are caused just by bad luck. Here's some examples. We've talked a lot about these: cigarette smoke, eating burned meat, BPA are things added to plastics, diseases and viruses, so on and so forth. We've talked a lot about these. All right, review questions. What are the 3 major types of mutations? What is a frameshift mutation? What are the 2 types? And what is the difference between a gain of function or loss of function? And what are the 2 major causes of gene mutations, which we talked about right here? Bad luck or mutagens? Let me know if you have questions. Thanks for watching. Have a great rest of your day.